So far in biology, we have talked about the submicroscopic level of atoms, molecules, and macromolecules, and now we're going to see how they fit into the simplest living thing, which is the cell. The cell is the building block of all life. In fact, it's the smallest entity that exhibits all the characteristics of life that we had talked about, which remember include order, regulation, growth and development, energy processing, responding to the environment, reproduction, and evolution. So every organism, every biological organism is made up of cells. Biological life can come in two different ways. You're either a single-celled organism like bacteria or protists, or you are an organism made up of two or more multiple cells, like plants, animals, and most fungi. So we can see that the simplest living organisms are just going to be one cell in structure, and the most complex are going to have millions and trillions of cells in them. Now, before we get into the inner workings of a cell, we're going to talk about microscopes, because if it weren't for microscopes, we wouldn't be able to study cells. So we usually use light microscopes in which we can increase the magnification on something. So if something's really, really tiny, we can use a microscope to make it look larger to us so that we can actually see the bits and pieces inside. Now there's two features of a microscope: magnification and resolving power. And magnification is basically increasing an object's appearance and size to its actual size. So making something that is actually really tiny appear really large. And then resolving power, this is kind of a weird one, but it's the ability of the instrument to show two objects as separate. So the better resolving power of a microscope, the clearer the image is going to be. You can definitely tell two tiny points of the cell as different from each other. If the microscope you're using does not have good resolving power, it's going to look blurry and you're not going to be able to see as many little detailed parts as if it had a better resolving power. If we take a look at this picture on the bottom here, this is an image, a typical image that we can see with a light microscope. So it allows us to see individual cells, which if you would look at some blood or your arm or whatever, you couldn't actually see the individual cells making you up. But if you look at it, you can't really see inside the cell. You can't see all the little organelles and stuff like that. So scientists have enhanced microscopes so that we can actually get a clear picture of what's happening inside the cell. These enhanced microscopes are called electron microscopes. So they kind of even dig and magnify deeper than just a light microscope. So they've greatly expanded the knowledge of our cells' interior and exterior. They can reveal organelles inside and outside the cell. And there's two different types. There's a scanning electron microscope and a transmission electron microscope. Now, just like their name kind of eludes, a scanning electron microscope uses a focused beam of electrons to kind of scan the outside surface of cells, so we can kind of see the characteristics of the outside of a cell, like we see in the picture here. Whereas the transmission electron microscope actually penetrates electrons through a cell so that they bounce off different inner parts of the cell so that we can see and get images of actually inside of the cell, like the organelles and such. So we as biologists really do have to thank the early microscope developers, Zacharias, Janssen, and Anthony von Leeuwenhoek and Galileo and all that, for messing with lenses to see what happens, because if they hadn't done that, we couldn't study the cell. And speaking of cells, let's get a move on with them. There are two major categories of cells. There's prokaryotic cells, which are the cells of bacteria and the archaea domain that we talked about in Lecture 1. And then there's also eukaryotic cells, which are the cells of protists, plants, fungi, and animals, including humans. So let's take a look at the difference between prokaryotic and eukaryotic cells. We are going to start our study with prokaryotic cells, since they are much smaller and older than eukaryotic cells. In fact, some scientists believe that the first prokaryotic cells appeared about 3.5 billion years ago. Now, prokaryotic cells are much simpler than eukaryotic cells. They lack internal structures that are surrounded by membranes. So they have some organelles, but they're not going to have as many, and these organelles are not surrounded by membranes. They don't have a nucleus to keep their DNA all together, and although they have functionally distinct regions, they don't have specific compartments like we'll see in eukaryotic cells. So if we take a look at this picture here, we can see that this prokaryotic cell, contains some organelles that we'll see in the eukaryotic cell, like DNA, although it's not in a nucleus. They have ribosomes and a plasma membrane, a cell wall, pili, which is kind of like cilia, and flagella. Most also have a capsule, I guess, around their cell wall as well. So they're very much fortified in a sense of not just having a cell membrane, but a cell wall and a capsule. Eukaryotic cells, on the other hand, are much more complicated than prokaryotic cells. There's three main parts to a eukaryotic cell, and here over on the right you see an example of a eukaryotic animal cell. Now, the three main parts consist of the cell membrane, which is a barrier. It keeps all of the organelles inside and happy inside the cell, and then it keeps everything else out of the cell. We also have the nucleus, which is the membrane that surrounds the DNA, which is inside the cell, so it keeps it nice and protected and together. And then we've got the cytoplasm, which is basically kind of between the nucleus and the cell membrane, and it's full of this fluid called cytosol. And in that cytosol, we have all the different kinds of organelles floating and working around in the eukaryotic cell. We will soon find out that there are actually two types of eukaryotic cells. There are animal cells and there are plant cells. And although they share a lot of different features, there's a distinct difference between animal cells and plant cells. So in this picture, this is a picture of an animal cell, and you can see the plasma membrane, mitochondria, nucleus, and all the different organelles inside there. And that is going to look different from this plant cell. Now, like I said before, animal cells and plant cells share most organelles. They're kind of the same. We still have cytoskeleton and mitochondria, ribosomes in the nucleus. We still have the three main parts, just like in the animal cell. We've got the cell membrane, the nucleus, and the cytoplasm. But there are a few organelles that are specific to plant cells. And these organelles are the cell wall, which is kind of like a fortified wall around the nucleus, the chloroplasts, which we will study when we study photosynthesis, and what's called a central vacuole, which is like a huge dumping ground for plant cells. So these organelles that are only found in plant cells and not in animal cells are right here. So we see this orange cell wall. We see the chloroplasts with the green stuff in it to represent all the chloroplasts, and then this huge, gigantic dumping bin of the central vacuole. So now we're going to start our tour of the cell. We're going to take a look at each individual organelle and see its structure and its function. We're going to start with the nucleus. The nucleus is the organelle that contains the DNA. Remember, DNA has genes in it. Specific sequences of DNA are called genes, and those code for particular proteins that control, the cells and everything about an organism. So the DNA is hugely important for cells. So it is bordered by what we call a nuclear membrane. It's a double membrane, and it's got little holes in it so that it lets RNA out, okay, so that they can make the proteins outside of the nucleus. So we need to have some movement in and out of the nucleus so that, you know, whatever's in the DNA inside can be translated to proteins on the outside in the cytosol. Now, interestingly, DNA, if we actually stretched out DNA, it would be a very, very, very tiny thread that would actually be about 6 feet long. That's how huge DNA, you know, would be if it were, you know, just one single straight line. However, obviously, our DNA is packaged so well that it fits inside the nucleus of a tiny, tiny cell that we can't even see with our own eyes. So how does this happen? What happens is that the DNA is covered with proteins, and these proteins are going to kind of wind the DNA up so tightly in what's called chromatin or chromosomes. And humans have 46 chromosomes, or packages of individual DNA molecules that are inside the nucleus. So you can imagine that each chromosome contains really, really long strands of DNA that are packaged so well that we can't even see them, with our eyes, and we can't see them with a light microscope. We have to see them with an electron microscope. So that is amazing. So you would think the nucleus would be jam-packed enough with 46 chromosomes inside, but we also have what's called the nucleolus inside the nuclear membrane as well, which is like a mass of fibers that produces the RNA that is needed for ribosomes to make proteins. So that's also inside the nucleus. So let's talk about that RNA and ribosomes next. Ribosomes are very small organelles that are inside the cytoplasm. There's a ton of them there. They're made up of 2 components. We've got RNA, which is one component of ribosomes. Remember, that's made in the nucleus, in the nucleolus of the nucleus. And then RNA also has a part of it that's protein. And the proteins are made in the cytoplasm. So the ribosomes come together and are made, assembled in the cytoplasm. The RNA kind of pokes out through the pores in the nuclear envelope and hook up with the proteins in the cytoplasm to make ribosomes. And the function of ribosomes is to make all of the cell's proteins. So ribosomes make the proteins. And like I mentioned before, there are tons and tons of ribosomes inside the cytoplasm of the cell. Some of them are what we call free ribosomes, and some of them are bound to a membrane or stuck on membranes of other organelles. So the free ribosomes, those that are not attached to proteins, are in the cytoplasm. So the free ones are in the cytoplasm, and they're going to make all the proteins that stay dissolved in the cytoplasm. However, the membrane-bound ones, the ones that are on other organelles, are making proteins that are going to be either incorporated into the membranes of the organelles, or perhaps the plasma membrane, or the proteins that are going to be secreted from the cell and go other places in the organism. So we have to definitely make that distinction between free ribosomes that are making the proteins, kind of for the cells that's going to stay inside the cytoplasm of the cell, versus the membrane-bound ribosomes that are going to make the proteins and package them for either incorporation in the membrane or to be secreted outside of the cell. Now we're going to go into much more detail about this when we talk about DNA structure and function and how proteins and stuff are made. But let's take a quick preview about how DNA controls the cell. So, like I said, DNA is always packaged inside the nucleus. It never comes out of the nucleus. But we do have little holes in the nuclear membrane for RNA to come out. So DNA is what we call translated into RNA, and this is what we call messenger RNA or mRNA. And the messenger RNA goes out of those little pores into the cytoplasm, and it goes to ribosomes, where it directs the synthesis of all kinds of different proteins. And we can see that here on the picture on the right. So we've got the DNA inside the cell. It's going to make the mRNA, which is going to go out of the cell to a ribosome to make proteins. So as mentioned earlier, outside of the nucleus we have the cytoplasm. And the cytoplasm is partitioned off in some places by membranes. And this is what we call the endomembrane system. And there are many organelles that are part of the endomembrane system. We have the endoplasmic reticulum here and here. So all this, okay, kind of that you see being partitioned off. There are the GOLGI apparatus, okay, which is this kind of wiggly membrane partition, and the lysosomes and vacuoles, okay, that are also bound by membranes. So let's take a look at each one of these in the endomembrane system. We will start with the ER, and no, not the emergency room, but the endoplasmic reticulum. This structure is present just outside of the nuclear envelope or the nuclear membrane. And there's actually two distinct types of endoplasmic reticulum. There's the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And they're connected, but they have different structures and functions. The rough endoplasmic reticulum is called rough because it has ribosomes that are attached to the outside of the membrane. So it's kind of dotted or speckled and looks kind of rough. Now the rough endoplasmic reticulum has the responsibility of making proteins, and it produces two main types of proteins. Proteins that will either be incorporated into the cell membrane or proteins that will be secreted from the cell. Okay, so if any protein is to be put inside the cell membrane, or if it's supposed to be spit out of the cell, it's going to be made in the rough endoplasmic reticulum. Now that is in contrast to what we call the smooth endoplasmic reticulum. And this is called smooth because it doesn't have any ribosomes attached to the outside of its membrane. It does have lots of proteins built into the membrane, so it's got a variety of different functions. The smooth endoplasmic reticulum can make lipids and steroids. It can, kind of detoxify the cell of drugs and poisons. And in fact, it's interesting to note that if a cell has an increased smooth endoplasmic reticulum, so it's got more smooth endoplasmic reticulum than other cells, then that can lead to drug tolerance because it's good at detoxifying drugs. So people who have an increased smooth endoplasmic reticulum count in their cells will have a higher drug tolerance than those that have lower amounts of smooth endoplasmic reticulum. So just kind of a little side note there. And one other thing also to point out, we see in the pictures on the right that there's kind of like a cartoon version of what we think the organelles look like. And then there's also the transmission electron microscope version, so kind of like the real picture of what we would see in a microscope of how these look. So if you take a look, the rough endoplasmic reticulum is right over here, and it looks, darker and kind of rougher around the edges, versus the smooth endoplasmic reticulum kind of just looks like these little bubbles in essence, and they're not, as rough on the outside, so they're more smooth in shape. And we can see that difference using a transmission electron microscope. So now let's move on to another part of the endomembrane system called the GOLGI apparatus. And the GOLGI apparatus's function is to refine, to store, and to ship chemical products of the cell. So it works really closely with the endoplasmic reticulum. In fact, products made in the endoplasmic reticulum are usually kind of shipped over to the GOLGI apparatus via transport vesicles. It all sounds so space-like. It's like, I can just imagine, the little proteins hop in the transport vesicle and over to, the Golgi apparatus. Now the Golgi apparatus is going to modify these, so they'll add functional groups here or there, maybe twist the arrangement of the particular product in order for shipping outside of the cell, or to store it in the cell somewhere else. Now after the Golgi apparatus kind of modifies any product that they've gotten from the endoplasmic reticulum, it also chemically tags the proteins for their final destination. So it's almost kind of like the UPS guy too. They put the little tag on it to say this is where you send it to. So the Golgi apparatus will package and tag the protein products, so that it'll be either distributed to other cell organelles inside the cell, or for transport outside of the cell. So the cell is just like this big working city almost, in essence, and it's really kind of cool. Another organelle that is included in the endomembrane system are the lysosomes, and they are membrane-enclosed sacs of digestive enzymes. And so we definitely want to make sure that they are membrane-enclosed, so those digestive enzymes just don't spill out in the cell for no reason and then just kind of kill all the cell parts. Now, the lysosomes are usually absent from most plant cells. That's what the central vacuole is kind of for in plant cells. And these digestive enzymes have several functions. They might digest food that is been stored in the vacuoles. They might digest foreign matter, such as bacteria that have invaded the cell. Or if a damaged, or if an organelle has been damaged or is no longer functioning, the lysosomes are going to, kind of suck it up and digest that as well. So these guys are almost kind of like the garbage men of the cell. Now, unfortunately for some, they have genetic mutations that lead to hereditary disorders called lysosomal storage diseases. So these are, abnormal lysosomes that developed. Some lysosomes will lack digestive enzymes, so they're not able to digest, food or bacteria or anything like that. The substances they can't digest will build up and then start to damage the cell and get too full and burst the cell eventually. One of these diseases is called Tay-Sachs disease, which specifically affects the nervous system or the brain cells. Tay-Sachs disease is found most prevalent in Jewish populations, although that doesn't mean it can't happen to anybody. In fact, it's basically it kind of comes on at around age 6 months is when you start seeing the effects of those substances being built up in the nervous system cells and starting to cause damage. And usually, unfortunately, it has a fatal effect by age 4, usually. So, in fact, if you ever have children in the future, your doctor usually, asks you if anybody in your family is Jewish or whatever, and if you've got that kind of, they will test for that Tay-Sachs disease gene if you show a strong prevalence for it. The last membrane-bound organelle, part of the endomembrane system, is the vacuoles. These are membranous sacs that usually kind of bud off of the endoplasmic reticulum, or maybe the Golgi apparatus or the plasma membrane. They're different sizes, they have different functions, but they're usually kind of the storage containers inside the cell. If the cell doesn't need something right now, it needs to store it away for either later use or for to break it down and digest it later. It goes into what's called a vacuole. So there's food vacuoles, there's contractile water vacuoles that store water, the plant central vacuole that plant cells have, kind of are just storage bins for stuff inside the cell. To take a little further look at the plant central vacuole, it's actually very versatile. It's huge, so it's very versatile. It can store proteins, within the cells of the seed. It can store water. It can store different pigments. It is just hugely versatile little organelle there that takes up a lot of space. So to review the endomembrane system, we get proteins being made in the rough endoplasmic reticulum, and then they, whatever product is made at the end kind of pinches off into little transport vesicles, and they go to the Golgi apparatus, where they get modified and they get chemically tagged for where they're going to go. So from the Golgi apparatus, they could either be stored in vacuoles, they could be, going to lysosomes, or they can be tagged to go be spit out of the cell or transported out of the cell, which are called, secretory proteins. So there's, it's a huge system that kind of packages everything and sends it all away to wherever it needs to go. Okay, so let's get out of the endomembrane system and take a look at chloroplasts. Chloroplasts are unique to plants and some protists, and the function of a chloroplast is to carry out photosynthesis. And photosynthesis is the conversion of sunlight into chemical energy of sugar. So it turns sunlight into sugar, and from the sugar or the glucose molecules, the plants can put them together in different polymers to make other organic molecules. So our chloroplasts are going to be kind of our energy conversion organelles, going to convert light energy into chemical sugar energy. The chloroplast structure is actually quite complex. The internal membranes are going to partition the organelle into three main compartments. The first compartment is found in between the two outer membranes. So in this space here is one compartment where chemical reactions can take place. The second compartment is called the stroma, which is inside the inner membrane. And then the third compartment is here inside of what are called grana, these stacks of tubes and sacs. These guys are called grana, okay, and that's kind of the third internal compartment. And it's inside of those little grana stacks where photosynthesis occurs. So in plants, the energy conversion from sunlight into chemical energy is made in the chloroplasts. However, we animals don't have chloroplasts, but we need to convert energy. So we have what are called mitochondria, and these are the sites of what's called cellular respiration that you're going to learn all about in a couple of weeks. Basically, mitochondria are going to harvest energy from sugars and other foods and convert it into chemical energy that the cell can actually use, and that chemical energy is called ATP, adenosine triphosphate. And the cells are going to use ATP as a direct energy source. So that's actually the packaging of energy that cells use in order to function. Mitochondria are found in almost all eukaryotic cells, including plants and animals. I mean, plants do have to use some of their sugars that they make in order to keep their cells alive. So they also have mitochondria. Now, the mitochondria are going to have two membranes that enclose the organelle, like the chloroplast had two membranes. The inner membrane has lots of folds called cristae, and these folds are going to increase the surface area in which ATP can be made. So there's tons of ATP because of all the little folds in the large surface area. There's tons of ATP that can be made inside of a mitochondria. And we're going to see in later lectures that, inside of these folds and membranes, there's all kinds of molecules and proteins that are needed for the cellular respiration. So that all happens on the inner membrane here. So this is kind of where the site of cellular respiration occurs, inside those little folds or cristae on the inner membrane. Next on the list is the cytoskeleton, and its function is to keep the shape of the cell and to help the cell move around. And the way it does that is through a network of fibers, which are made of proteins, and they're kind of extending all along through the cytoplasm. Okay, and they're going to act like the skeleton and muscles of the cell. So it helps to support it and keep its shape, not kind of like all blobby, but firm shape. And then these little fibers can detach themselves and move the cell with it. It's kind of cool how it works. So here we're going to see what the cytoskeleton is actually made of. So there's three main types of fibers that are responsible for the skeleton and movement of cells. We have microtubules that are made from the protein called tubulin. So those two words are kind of related in a way. There's microfilaments, which are made from the protein actin. And then there's also what we call intermediate filaments, which are made from all kinds of different proteins. But then two major ones that you'll need to remember are the tubulin and the actin. The tubulin are the microtubules, and the microfilaments are made of actin. Tubulin is the bigger protein, and the actin is the smaller protein that kind of walks over the microtubules in order to kind of make the cell move. It's pretty cool. The next organelle that we're going to talk about is one that not all cells have. It's called flagellum. A flagella is an appendage, basically, to a cell that helps it move around its environment. So it propels the cell by undulating whip-like action. So it's kind of like a propeller in a sense. Now, in humans, the sperm cell is the only cell that has a flagellum. All the other cells move by the cytoskeleton rather than the flagellum. All right, the last organelle that we are going to talk about in this lecture is called cilia. And it's kind of related to flagella, except they're shorter and more numerous. Usually a cell with flagella will have one or maybe just a few of them. Cilia, there are lots of them kind of all over the surface of the cell membrane. And these guys are also, like flagella, going to promote movement by kind of coordinating back and forth motion of these things, like so that they'll kind of, it's like little hairs that sweep in the same direction to move the cell. So the cilia can either promote actual cell movement, or like in our intestines, they can be present on non-moving cells, but their function then would be to move fluid kind of down and across the cell membrane. So that's like in our intestines, we have cilia on the cells of our intestines that move those digestive juices kind of down the intestines and move the nutrients towards the cell membrane so that it can absorb those nutrients and get into your body. Okay, so that's all of the organelles that we're going to talk about in this lecture. In the next lecture, we're going to spend most of the time talking about the organelle called the plasma membrane, because it's very complex in its structure and its function. It's very diverse, kind of in what it does. So we'll save that one for the next lecture.